Okay guys, so as I promised, I think it was like a week ago, maybe a little longer, when I started working on the color photo series regarding African cichlids, although this does apply to other fish as well. I did say that I was going to work on like a series of these. Today we're actually going to be talking about the color blue. So just to recap real quick, we all learned this in science and in art when we were in school. There's color that we see, but how we get that color and how we see it really depends on pigment and light, the combination of the two. And there's different effects that happen in order to create what we visualize, especially within the animal kingdom. Fish are very multi-dimensional. So today, just to recap that really quick, when we're talking about light, we have blue light, red light, and green light color within a spectrum, okay, within the rainbow. They mix to produce white. When we're talking about pigment, we have blue pigment, red pigment, and yellow pigment, and all other colors are a combination of that. And if we combine all three of these just right, we'll end up with black, because as pigment concentrates, that's what we move towards. That's just a little bit of a recap and a preface before we dive into what we're going to be talking about today. Today we are actually going to be talking about blue color in African cichlids. So let's clear this. As I said last week, and this is kind of mind-blowing for a lot of people, there is no actual blue pigment in African cichlids. There are very few animals or anything in nature that actually has a naturally blue color. It's usually some kind of trick of the light. For example, I talked about this in the last video, when we are looking at a blue jay, the feathers aren't blue. They have literally little light bending portions of their feathers that reflect the blue range of white light, and that is what our minds interpret as blue. It is reflecting the actual blue light from the spectrum. Similarly, this also happens in African cichlids. There are no cyanophores in African cichlids, or any cichlids for that matter. Very few fish in the world actually possess cyanophores, which is blue pigment cells. So how do we get blue? Well, it really depends on which kind of blue we're talking about, because we have to think of color with these fish almost as if we're looking at a type of artwork that's layered, okay? Think of them like art. And they have different dermal layers, as I was talking about last week. So we have about three, and you know, just like we have our outer layer of skin, dermis, epidermis, all that, and it goes deeper and deeper and deeper. We're also, to quote John Leguizamo in one movie, a translucent biped. So our color and our pigment of our skin is determined by the pigment in the different layers of our skin. The same thing happens with fish. So we have this deep dermal layer that's like pretty much almost towards the flesh itself. Then there's like another layer on top of that where oftentimes reds and yellows are going to be held. And then even on top of that, and sometimes there's going to be darker pigment in here, sometimes there's melanin in there. And then we have the outer layer that's right next to the scales, and very little color pigment-wise is actually held there, but an important part of how we see color is in fact deposited there. That's right next to the scale layer. So in this outermost layer, we actually have what are called iridophores, okay? These are the cells that do produce a visualization of color, but they don't possess pigment. Rather, they actually possess crystals, guanine crystals, and these crystals bend light, specifically blue spectrum light, kind of like looking at a moonstone if you've ever seen one. It has, like, as you tilt it in the light, it sheens this ultra blue color. Or again, if you look at blue jays, their feathers reflect blue light. It's the same exact concept. When this happens, we actually have an overlay, okay? So when you have the dark melanistic layer that's further toward the back, and then we overlay it with that crystalline layer that has iridophores, we end up with a blue effect. Okay, I don't know if y'all can see that very well, but and we have different saturations of blue, of course, depending on how much melanin we have will determine the severity or the depth of blue. So in the case of this like very dark melanistic fish that has black pigment, melanin, in the lower dermal layer, overlaying that here on top of it, if our eyes are seeing this way in, so this is the way we're looking at it, we're actually looking at an effect created by a translucent layer that has the quinine crystals that are reflecting blue light. They are backed by black melanin, dark melanin. And when that happens, we end up with different saturations of blue. So against a very dark melanistic fish, we can end up with a deep blue. When we are talking about lighter fish, well, let's do this. When we talk about slightly lighter fish, it can be a lighter blue color, and so on and so forth. Now, the interesting thing here is that when we start combining other pigments, we get different effects because, as we all learned in art class, you mix yellow, red, blue to get different colors. So if we mix yellow and blue, we get green, right? So if we happen to have a fish that has, say, melanophores, but then also has some xanthophores, which are the yellow color, we end up with kind of a yellowish green, okay? And that's the effect that is created. But still, it's all dependent on multiple pigments playing in conjunction. So, for example, let's talk about iceberg friar eye. That's one of the greatest examples I can give. I started to talk about it last week, was getting a little long on time. So with iceberg friar eye, we start with this fish that has melanin almost all the way across the body, and as they're young, they start to actually drop melanin in the top of the dorsal and the top area of the body. Just like octopi, these fish in mood and dominance and whatnot are able to change how they are expressing some of their pigment cells. So they're actually dropping in dominance some of their heavier melanin in the top of the body, and they're actually able to drop some of it all the way out, which is why it appears so white across the top of the fish. Because when they're expressing that way, not only are they increasing the amount of iridophores, not only are they increasing the amount of iridophores in this outside layer through their dominance and depositing more quinine crystals in that outer layer, but simultaneously they're dropping out some of the melanin in these layers. And in fact, in the top part of the body, you'll see some that they call ghost iceberg, where the body's nearly all white. They're actually dropping all of it out. Now, I've actually seen some very beautiful iceberg friar eye then end up getting dominated by another male fish of the same species, and the melanin will come back. All of a sudden, they won't have as much blaze anymore. And it's literally that they're putting melanin back into that dermal layer and creating the effect again of having, of having the dark top of the body. But the takeaway from this is to understand that that top half of the body that looks very white, but it's kind of powder blue, that is exclusively because there's no melanin behind it, okay? The front part of the cellular layer, of course, is reflecting white to white blue light, and when it's backed with no melanin, but rather just whitish flesh, it's going to appear white. The lower half of the body still does possess the melanin. So when we put that same crystalline layer on top of it, we get a dark blue effect. And this goes across many different species. Even if you want to talk about, say, star sapphire, okay? Phoenochilus, Tanzania. Lupingu is one of the best locations for those, actually. But they show a beautiful, deep, rich blue in the wild. And then as they start to mature, they start to get the chips. And those chips are localized concentrations of the guanine crystals. And those crystals actually grow and create larger and larger chips. If you've ever seen a progressive photo sequence of a development of one of those star sapphires, you'll notice how they'll start with like a little chip somewhere, and then by the time that they're fully developed, the first chips that they got will usually kind of grow and become larger. So it's very interesting. I don't want to go too far past this today just because I don't want to get too much deeper into things. Once we start talking about reds and yellows and really get into mixing a lot of other colors, it'll be easier to understand some of it. But I thought that the best way to start would be really to dive into blue specifically, so we could start understanding how layering one thing behind another creates an effect. And that effect can be best represented by this little black dot here. Oops, sorry. This little black dot here with the blue chips on top of it, okay? So when we're talking about these fish, again, there's no blue pigment in the lake. There is blue visualization, and that comes in a myriad of different effects, colors, intensities, etc., etc. So remember, the more dark color melanin that we have behind the iridophores, the darker the blue. The lighter the blue effect, that means that there's less melanin back there. And the higher the concentration of the reflective aspect of the fish, the iridophores, is going to create a greater intensity in shine, because it is a crystallized iridescent layer. It's a reflective layer, as well as being able to cover up more and more of the pigment behind it. Because if you're able to reflect 100% of light, you're able to just look white even if there's something behind it. Although in the case of cichlids, more often than not, when we're seeing truly white fish, it's because either the fish doesn't possess melanin in the dermal layer to begin with, or they're able to actually shut off part of the melanin in the dermal layer, such as in the case of the iceberg fry rye. So I hope this is informative for you guys. I hope that it's easy to understand. Y'all are welcome to give me any kind of feedback or ask any kind of questions. I'll get furthermore details with you guys, and we'll go from there. Probably next week I'll be looking at working on either yellow or red. Red's going to be kind of a controversial topic because we're going to get into a few different things about how it's possible to affect that coloration via what we feed the fish. And we will have a whole video talking about food, as well as lighting, all this type of stuff. But as far as purely what the fish do within their own genetics, within their own development, and what their bodies are able to produce visually, this is how they do it for blue. So I hope you guys enjoy it, and I'll see y'all next time.